Hello students, welcome to chapter 10. We're going to be talking about cell division and mitosis. So mitosis is in of itself the splitting of cells. Cytokinesis can happen without mitosis, but it's going to cause problems if you don't have mitosis with cytokinesis. Okay. Really quick, we're going to talk about the DNA itself. So first we have DNA in its packaged form, which is chromosomes. So DNA itself is very, very long, right? Without it being packaged up within chromosomes, it would have a hard time fitting inside the cell. And within the nucleus, it's mostly going to be packaged up, right? So when it's getting ready to split, it has to be packaged up completely inside the chromosome, inside that, what you see is an X format, that chromosome format. So DNA fits into a nucleus because it's packed into shorter length by histone proteins. And those are those little round things that you see the DNA fibers wrapped up into. What you would see as like a spool. If you don't know what a spool is, that's what fiber is wrapped around whenever you see it in stores. Other non-histone proteins associated with DNA, the complex of DNA and all its associated proteins is chromatin. So any other type of proteins or stuff involved with DNA is called chromatin. In a nucleosome, an 8-protein nucleosome core particle forms when DNA winds around these 4 histone proteins. So you have a nucleosome, basically a bound together DNA particle, which you would say a particle wound into those histone proteins. A shorter linker segment of DNA connects those nucleosomes. Chromatin packing continues at higher levels, with euchromatin packing loosely together and heterochromatin packing densely together. So the euchromatin is loosely packed together, which means that it can genetically be activated by other types of proteins. And then heterochromatin is more densely packed together by these histones. They'll really wind it together by those histones. Heterochromatin is less genetically active or even not genetically active. Sometimes they'll just sit there in the DNA and not actually do anything. So let's say that some of the cells inside your head don't account for the same type of DNA as some of the cells inside your legs, right? So you don't want the same type of genetic code to recognize like leg cells inside your head, right? So it's not going to grow a leg out of your face. So chromosomes themselves are nuclear units of genetic information divided and distributed by mitotic cell division. So basically just pack together DNA, and when they split apart, they will split apart evenly, and they'll be exactly the same. The number of chromosome sets is called its ploidy. Ploidy. Okay? Anytime you see the word ploid at the end of a word, it's ploidy. Okay, so if it's haploid, it'll have one set of DNA. If it's diploid, it'll have two sets of DNA. Okay, haploid can also be said in the same terms as monoploid. You can kind of interchange that, but just call it haploid for right now. Monoploid also inters that diploid was involved and you split it into two, and there's lots of different variations involved with that. So just say haploid is 1 and diploid is 2. So many plant species have different versions of that. So you can have 3 or 4 or even 8 or 16 or 24. I mean, there's tons of these. But in animals, that would be lethal. Okay. Part of the reason for that being is because plant species have a very wide variety of polymorphic traits. Okay, so fruits, if you ever see a crab apple, crab apples are actually that small. Okay, they're very tiny. The apples that you see at the store are like hand fruits. You can pick them up with your hand. Those types of fruits are actually polyploidy. Poly meaning many. So polyploidy apples, those are actually genetically inclined to have multiple sets of DNA. Crab apples are usually diploid. The reason why they're grown like that is because of market cells. Watermelons in the wild are usually only about that big. Watermelons that you buy at the store are usually 16 ploid. You know, so these are bred to have multiple sets of chromosomes, and they're sold as that. The main reason for this is because of market sales. But plants can handle this. Animals cannot. You get one set of chromosome more, and you get things like Down syndrome or gigantism, or, you know, you get problems with the animal. So, yeah, you get some problems. Anyway, DNA synthesis replicates DNA chromosomes into 2 copies with almost perfect fidelity. I say almost perfect fidelity because there can be mutations, there can be other problems that involved, and we can talk about those a little bit later. The 2 chromosomes of each pair in diploid cells are called homologous chromosomes. Homologous meaning same pair, same logic, right? Same set. The same genes in the same order. So this is an overview of the entire cell cycle. In multicellular eukaryotes, cell division is under strict control. The life cycle of a cell, or the cell cycle, is divided into 3 different parts. And we have mitosis, we have interphase, and we have cytokinesis. So let's start off with interphase. Interphase is divided into 3 phases. First, we have the gap 1 phase, or what we call G1. G1, the cell grows to twice its size. Organelles will double in number, proteins and lipids double in amount. Right? So this is important because it needs to be able to have the same amount of proteins, the same amount of lipids, the same amount of fluid inside of the cell whenever it starts splitting apart. Right? So if this one doesn't have enough proteins and this one doesn't have enough lipids in it, it's going to be much smaller than this one over here. Right? And it's not going to be able to function that well. The next phase is synthesis phase. This one may take a bit longer than G1 because it's replicating the DNA. So each of its 46 chromosomes is duplicated by the cell, right? So each of our 23 pairs is duplicated. And then the next phase is gap 2 phase. More growth of the molecules, including microtubules. Okay, this is where it starts to grow those microtubules. G1 is also the stage in which many cell types stop dividing. Okay, at some point, there's some cells that will stop dividing after cytokinesis. In some cells, it'll go into a G0 phase, right? And then it'll just stop dividing at that point. It'll stop growing, it'll stop dividing, it'll just stay as a cell. And this happens sometimes just because, like, all of the other cells around it are perfectly fine. Right? Think about your skin. If your skin is perfectly fine by itself, it's no cuts, no anything like that, you don't need to grow more cells on top of your skin. Or let's say your heart, right? If your heart is perfectly fine at that layer, you don't need to grow more cells at that layer. That being said, I'm not a doctor, I don't know all this stuff. But if your skin cells at that layer is perfectly fine, it doesn't need to grow more cells at that layer. Granted, your skin flakes off all the time, but it'll grow more cells underneath that layer. Internal regulation controls each phase of the cell cycle and regulates the overall number of cycles that a cell goes through. Okay, and this is important because there are checkpoints that interphase must go through in order to get through every single phase, so G1, S, and G2, just to get through and get to mitosis. And we'll get to those in a second. So, before a cell divides in mitosis, duplication of each chromosome and its proteins produces 2 identical copies, right? And this is in S phase, and they're called sister chromatids. So each of those 46 chromosomes has to produce an identical copy of itself. Sister chromatids are held together by sister chromatid cohesion until mitosis separates them. And this is usually just by a hydrogen bond. Cohesion holds the sister chromatids together until they're removed. And they're removed by those spindle fibers, right? And they'll just pop them apart. The equal distribution of chromosomes into each of the daughter nuclei is called chromosome segregation. So, you have sister chromatids that are together. Once they're apart, they're called chromosomes. Let's go ahead and start into mitosis. So the first stage of mitosis is prophase. Chromosomes condense into chromatin. The nucleus becomes smaller and it starts to disappear. Right? The nucleolus becomes smaller and it starts to disappear. That nuclear membrane starts to disappear as well, right? And so it starts to degrade. The mitotic spindle begins to form between the two centrosomes as they migrate towards the opposite ends of the cell, where they'll form the spindle poles. Okay, so they're starting to come in to try and get to those centromeres. Prometaphase is the next step. It begins when the nuclear envelope is totally broken down. Spindle microtubules grow from the centrosomes at the opposite poles towards the center of the cell. So they're more towards the center. A kinetochore forms at each of the centromeres on each of those sister chromatids. So kinetochores connect the points where those spindle fibers, right, connect to the sister chromatids. So we have the sister chromatid, it's going to connect where those where those spindle fibers connect. Kinetochore microtubules bind to the kinetochores, right? So it's going to bind to that. Non-kinetochore microtubules overlap those from the opposite spindle poles. So if you have another microtubule, it's just basically going to connect to each other. And that's just going to push each other apart. The next step we have is metaphase. Spindle microtubules move chromosomes into alignment at the spindle midpoint. What this is called is the metaphase plate. Now, there's a good reason why they want to put it at the midpoint. If they don't put it exactly at the midpoint, what can happen is parts will start moving into a different direction and then start leading other chromosomes towards that direction as well. If you put it to the midpoint, it'll split evenly. Condensation gives each chromosome a characteristic shape determined by length and centromere location. So each chromosome is set up so that there's a specific shape, there's a specific length, and there's a location involved with the centromere. Image of a complete set of metaphase chromosomes. They're all arranged to its set, its shape, and it forms a karyotype. Now the karyotype is every single chromosome within the cell. It's all arranged according to its karyotype. The spindle separates the sister chromatids and pulls them towards the opposite spindle poles. So this is anaphase. Ana meaning apart or separated. So anaphase, this is metaphase, this is anaphase. So movement continues until it's separated the chromosomes, breaking that hydrogen bond, moving the daughter chromosomes, having reached the two different poles. At this point, the chromosome segregation is complete. And then we move on to telophase. The spindles start dissembling at the chromosomes, and each spindle spindle pole decondenses, the nucleolus just reappears, RNA transcription starts resuming. We have this new nuclear envelope reappearing, right? Nuclear division is complete at this point. And what you can see in the center is a plate forming. Now that plate, or a fold in our case because we're animals, the plate is formed by plants, is brought on by cytokinesis. Mitosis can happen without cytokinesis. Cytokinesis can happen without mitosis. Most of the time, they're happening at the same time. So, cytokinesis produces two daughter cells, each one with two daughter nuclei most of the time. In animals, you have a contractile ring that's forming up between the two cells. That creates a cleavage furrow. Okay, that cleavage, it cleaves the two cells apart. In animals, it has a furrow girdling the cells and deepens until it cuts the plasma into two parts. In plants, it has a cell plate that forms between the two, dividing the cytoplasm, and eventually it'll form a cell wall and all the structures in between. The plane of cytoplasmic division is determined by the layer of microtubules, and the persists at the former spindle midpoint. So all of this is kind of going on at the same time. It's a flow. Okay, I want to say that this mitosis is a flow. It's not direct points of interaction. Okay, so prophase flows into metaphase, prophase flows into prometaphase, flows into metaphase, flows into anaphase, flows into telophase, right? And most often, cytokinesis will often start in the middle of anaphase, because that's where you'll see the either cleavage point or the cell plate forming. Formation and action of the mitotic spindle. So the mitotic spindle is made up of microtubules and motor proteins. So there's these motor proteins that literally walk along the microtubule. Approaching mitosis, microtubules reorganize into the spindle. This is basically how these chromosomes are being split apart by those microtubules. So the microtubules reorganize into the spindle, which fills almost the entire cell, right? So they'll fill up the entire cell. The centrosome is the main microtubule organizing center, which is called an MTOC. These are in animal cells, many protists. A lot of plants have a MTOC, but they don't have centrosomes. Centrosome contains a pair of centrioles. Microtubules extend from the centrosomes, produce star-like arrays, those asters, you can see them from either side, that form the poles of the spindles in prophase, or G2, depending on where it's starting from. Like I said, this is a flow, so you can't exactly determine either or. So this is kind of a representation of what's happening here. You can see here that along the kinetochore, so that's that purple part down below, is where the DNA is. That's the chromosome. And that's the spindle pole, the spindle fiber coming up along that edge, or along that rope looking thing. So the microtubule motor protein is literally pulling it along that kinetochore microtubule, that spindle fiber. And as it's pulling it along, it's degrading any more of that spindle fiber. So it's pulling it up as well as degrading that rope. So you remember those checkpoints that I was talking about before, those interphase checkpoints? There's also a metaphase checkpoint here. So in the cell cycle, we have 3 key checkpoints to prevent any phase disorientation. G1 to S checkpoint. This one checks for nutrients, any growth factors, or DNA damage. This one just makes sure that there's enough nutrients, that there's enough growth factors involved, or that there's not any DNA damage involved with this cell. If there's any type of problem with these, it's going to go into its resting state, or it's going to go into G0. Basically, that cell is just going to die, right? Because you don't want a cell that has DNA damage. You don't want a cell that's going to replicate that DNA, otherwise it could become cancerous. It could become something that you don't want. Okay, G2 to M, G2 to mitosis checkpoint. This one can check for cell size. It could also check to see if the DNA has been replicated. So, if the cell is not large enough, you don't want it to duplicate, right? So if it's not large enough, you don't want it to duplicate a new cell that's not large enough, because then you're going to have 2 really, really small cells. If it hasn't been replicated, if the DNA hasn't been replicated, you're going to have one cell that has one DNA and the other cell doesn't have any. So that's another option that could happen, and if it doesn't, then it could be that the cell is going to go under some type of dying process. Okay. Another checkpoint is the mitotic spindle checkpoint. This happens usually during anaphase or metaphase. So if there's some type of spindle attachment that didn't occur, let's say that the spindles came in and it didn't attach to a certain DNA chromosome, this could be a problem. Right, so proteins go in, they try to check to make sure everything's going right. If something's off, that's a checkpoint issue. And maybe it'll fix it, maybe it won't. If it doesn't fix it, the DNA is going to be wrong on one cell. The DNA is going to be wrong in the other cell. So these are issues that could happen inside the cell, and sometimes it fixes it, sometimes it doesn't. If that's a checkpoint issue that does occur, sometimes the cell gets messed up. So these are checkpoints that inactivates the cell cycle to proceed. It stops the cell cycle from proceeding at that point. And these are the types of proteins that stop those cell cycles from proceeding. Cyclin-dependent kinases. So direct regulation of the cell cycle itself involves internal control systems. And these are called cyclins. Enzymes called cyclin-dependent kinases, or CDKs. This is a protein kinase which phosphorylates and regulates the activity of target proteins. Basically, if you don't have these that are working right, you're not going to have the cell cycle working right. So M-cyclin CDK1 is a complex protein that regulates the M phase promoting factor. So this is the M checkpoint. So when a chromosome is currently attached to the mitotic spindle, MPF activates another enzyme, the anaphase promoting complex, or APC, and activates APC, degrades an inhibitor of anaphase, leading to separation of sister chromatids. Now there are external controls that modify internal controls. Signal molecules originating from the outside. These can include hormones, growth factors, UV light, X-ray radiation, any type of external control. Okay? Reactions triggered by the activated receptor may speed, slow, or stop the progress of cell division. So, first of all, when you get contact inhibition, cells basically stop dividing. Okay, this is something that we've known for a long time. This is something that cells just do. So contact inhibition triggers internal reaction pathways that inhibit division by arresting cell cycle, stabilizing cell growth in fully developed organs and tissues. Right? Just what as I was explaining, if you have all your skin in one tissue layer, it's not going to continue to grow out. Right? It's not going to continue to fold and fold and fold. You would have weird skin. Cells in contact with one another are shunted into the G0 phase. They prevent themselves from dividing over and over and over again. Right? So a new layer is going to find itself on below that layer, right? It's going to grow up above or below that layer, and then the next layer is going to below that, the next layer is going to below that, you know. If contacts are broken, the free cells often enter rounds of division. Right? So in case you cut yourself, you know, it'll grow something else in between that cut, but it's not going to grow, you know, through another layer, and it's not going to grow out. But let's say that this gene is off, that there's some type of gene that takes this gene into a different phase, and the G0 phase gene is off. This is called an oncogene. It'll continue to grow. Okay? And this is kind of what causes some types of cancer. Okay? So tumors can break loose and spread in what's called a process known as metastasis. So types of skin cancers can continue to grow from the skin. And it'll break off into your bloodstream, and it will break off into your, you know, body, and it can go into your system and continue to grow. It's a bad thing. My grandmother has it. So we talked about eukaryotic cell division. Cell division in bacteria goes through binary fission. Basically just by splitting themselves into two parts. Most prokaryotes have a single circular DNA molecule known as a prokaryotic chromosome. In E. coli cells, which are capable of dividing every 20 minutes, DNA replication occupies 19 of those minutes. So it takes a while for the DNA to replicate. And then once it's done replicating, split. So you can see over here on the side how the differences are. For your attendance assignment, what I'd like you to do is tell me a protein involved in DNA replication. Name me some protein involved with DNA replication. And that's going to be it for this round. I will see you later.